Hello, everyone, and welcome to today's workshop. Anna Paxad will be presenting this comprehensive workshop that features a presentation, a live microscope demonstration, including 4D STEM data acquisition and processing, and an open Q&A session. The microscope session will be performed on a STEM system equipped with a Stela hybrid pixel electron detector and a STEMx 4D STEM diffraction system. Anna got her PhD in mechanical engineering in 2011 and has been working at Gatan since. Throughout the last 10 years, she has worked on several projects developing SEM and TEM products for Gatan. She is now in charge of the diffraction product line, including the Stela camera and the STEMx system. And now for a little housekeeping. If you have any questions throughout the event, please type them into the questions panel on the GoToWebinar pane. If there is time at the end, we will answer some questions live. However, if we run long, Anna will address your questions offline within the week. And with that, I will hand it over to Anna.
Thank you, Parker. Hello, everyone, and welcome to our workshop today. That was a good introduction, Parker. We are going to, as you just heard, have a short presentation introducing the 4D STEM technique and the Stela camera, which is our newest camera. It's a hybrid pixel detection system that we are using for our workshop today. Then I will switch to the microscope computer and demonstrate data acquisition, 4D STEM data acquisition on the microscope with the Stela and the STEMx system. And then we will do some data processing within the digital micrograph. We have a lot to go over, so let's just start. So what is 4D STEM? Basically, this is a STEM diffraction technique where, in addition to acquiring the signal on a STEM detector, as you can see on top here, it could be different types of STEM detectors, we have a pixelated detector which is also collecting the diffraction pattern at each pixel position while you're scanning the sample. So you usually scan the surface of the specimen in a two-dimensional array of pixels, and you have a two-dimensional diffraction image at each pixel position as you're scanning the sample. That is why this technique is commonly known as 4D STEM, 2 dimensions on the scanning dimension on the spatial domain, and 2 dimensions on the diffraction domain. Now, for acquiring this type of data set, you obviously need a pixelated detector, and you need to be able to control the scanning unit on the microscope. And we do this with a product at Gatan called the STEMx. So STEMx allows you to do hardware synchronized 4D STEM diffraction data acquisition using any of our Gatan our cameras. So you can do this with the Rio IS, 1DO IS, K3 IS, and the Stela detector. And so we connect the STEMx box to these cameras, which allows you to do hardware synchronized data acquisition in digital micrograph. And once you collect your data, you can process the data depending on the type of experiment and sample that you're looking at. We have different tools in digital micrograph to do the data analysis, as we will see later on during the workshop. So how does all this work? As I said, this is done via hardware synchronization to the camera. You use digital micrograph interface to set up your experiment for data acquisition. So we have a technique in digital micrograph called SI acquisition technique, as you can see on the left-hand side here, where you select what type of signal you want to collect during the spectrum image data acquisition. In the case of 4D STEM, it will be seabed or diffraction pattern. At the same time, if you have other detectors, for example, a bright field, dark field detector, or an EDS detector, you can simultaneously collect the signal as you collect your diffraction data. In this panel, I will show you during the demo, you set up your data acquisition parameters such as number of pixels in your 2D array, the pixel dwell time, so that's how much exposure per diffraction pattern you want on each pixel, and several other parameters. And when you're ready, you click capture. When you click capture, what happens is that the pixelated detector, in this case, we have a Stela camera, for example, captures a diffraction image and sends it to the digital micrograph computer. If you have selected during your setup other signals, such as a dark field detector signal or EDS, those signals are also simultaneously captured. Once the diffraction pattern is collected, a signal goes through the STEMx box, as you can see here, to the DigiScan. And DigiScan is our scanning control unit. And then DigiScan tells the microscope to move to the next STEM pixel position. And this continues until your whole ROI is scanned and the whole data set is collected. So everything is set up and simply set up in digital micrograph, and then you don't need to do anything. We automatically collect the data via hardware synchronization of the scanning system with the camera. So once you have collected your 4D STEM data, as I said, there are different tools for you in digital micrograph without need of data conversion or transfer to process the data. We have tools for strain mapping, we have tools for visual imaging, and also differential phase contrast imaging. I will show you examples of all of these. And in addition to that, if there are further analysis or different types of analysis that you need to do and they do not exist in digital micrograph, you have the capability of writing either a DM script or a Python script in digital micrograph to further analyze your data. Here is, so I'm in the next few slides, I'm going to describe how we do some of these data processing in digital micrograph. So the first one I talked about is strain mapping. In order to do strain mapping, we collect nanobeam electron diffraction data for 4D STEM, and we compare the diffraction patterns in the region of the sample that we expect to have strain with a diffraction pattern from an unstrained region of the specimen. And by calculating or measuring the shift in the position of the diffraction reflections with respect to the reference diffraction pattern, we can calculate how much strain is in the sample, because the distance between diffraction spots is inversely related to the distance of the crystalline plane. So like I said, you collect a two-dimensional scan of the sample. At each pixel position, you have a diffraction pattern. In the software, you specify a region of the sample where you know there is no strain. In this case, this is the substrate, which we know there is no strain. This reference diffraction pattern can be from a different data set as long as the data acquisition parameters are exactly the same. It doesn't have to be from within the same data set that you're trying to analyze. In the reference diffraction pattern, you specify the crystalline directions for which you want to calculate the strain, and then software automatically calculates the strain for you in those directions. I will show you an example when we get to the data processing part. This is a nice paper that was published a few years ago, just to bring up the point that strain analysis with 4D STEM is not just limited to crystalline materials. You can also apply the same concept and map the strain in amorphous materials. So this was done by the group up at STEM. And again, so you collect 4D STEM data sets, you have a reference diffraction image, and you compare the areas in which you expect the strain. In this case, they were pulling on this sample and applying some load, and by measuring the change in your diffraction pattern, you can calculate the strain in your specimen. On the bottom right here, you can see the reference if you're interested to read more about this. The second type of application is virtual imaging. So let's see how this works. Again, you scan your sample in a two-dimensional array. At each pixel position, you collect a diffraction pattern, so you have your four-dimensional data cube. Now, if I look at my real space dimension here in panel A, and I apply in the software, let's say this square ROI, this would be as if I have a square selected area electron diffraction aperture. So the software goes and it sums the diffraction images within this selected region on the sample and gives me this diffraction pattern in panel B. So this is a virtual selected area diffraction pattern. And I can do this with different shapes and combinations of virtual selected area apertures and get different diffraction patterns this way. Now, on the other hand, if I go to the diffraction domain, I can do the same thing and design in the software my virtual objective apertures. So you can see, for example, here, all these selections, all the reflections in the blue circles. If I make this selection as my virtual objective aperture, what the software does is it goes in my four-dimensional data cube here. At each pixel position, looks at the intensities within the selected regions in the diffraction pattern in these blue circles, sums them up, and then assigns that number to that specific pixel. And that's how you generate your virtual dark field or bright field images. And I'll show you a couple of ways to do this in DM when we go to the software. You can get a little bit more fancy and advanced with this type of analysis and generate orientation maps of your samples. So here what you're doing is basically classifying your diffraction images and based on the orientation and different classes that you have in each pixel, you color code them and you get an orientation map. Again, if you're more interested to see this, there is a paper reference here. You can go and read more about this. And I will show you an example also in digital micrograph where we have done this via Python scripting in DM. The next one that I wanted to talk about is differential phase contrast imaging. Here, basically, the concept is that the electric or magnetic field in your specimen, in your thin specimen, causes a deflection in your electron beam. And if you measure that deflection, you can then directly correlate that to the electric field in your sample. So you collect a 4D STEM data set, and at each pixel position, you measure the position of the center of the mass and how much it's moving as you're scanning the surface of the sample. And that gives you this dx and dy maps. And then you can combine these two and calculate your beam deflection map, and from that, the divergence. So, and once you have, so like I said, the beam deflection is directly related to the electric field in your specimen. And once you have the electric field in the sample, you can calculate the local charge density in your specimen as well. And this is very nicely shown in this paper published from the group at UC Irvine using the OneView and the K2IS cameras a couple of years ago now. So as I said, you collect your 4D STEM data, you calculate the shift in the center mass or the beam momentum, and then from that you can calculate the divergence, which gives you the local charge density in your specimen. Okay, so this was the introduction part to 4D STEM. As I said, STEMx allows you to do hardware synchronized 4D STEM data acquisition with any of our cameras, including the Stela camera, which we're going to look at in more details today. And once you have your 4D STEM data, there's a lot of different types of data analysis, depending on how you set up your data acquisition and what type of sample you have that you can do with this type of data. Let me see. Okay, I'm pretty good on timing. All right, so what is Stela? Stela camera is our latest camera that we have announced for a few months now. It's a hybrid pixel electron detector, which is fully integrated in digital micrograph. That's Gatan software for electron microscopy data acquisition and data processing, and is the only solution that allows you to do this fully integrated in digital micrograph and collect 4D STEM data sets. So you get the highest quality diffraction data sets because it's a hybrid pixel electron detector, and it's the fastest way to result for you because it's fully integrated in Gatan digital micrograph. So let's look at a little bit more in details what we mean by highest quality diffraction data. So with the Stela camera, which is a hybrid pixel detector, you have a very high dynamic range. So you have the capability of capturing very weak and very high intensity signals in single frame. And it's a very fast camera. I will show you, we have different operating modes with the camera, and you can collect 4D STEM data cubes at more than 16,000 pixels per second. I'll talk about this in more detail, but if you remember, I said earlier, we make the camera the timing master. So since we can run the camera at 16,000 frames per second, we can collect our 4D STEM data sets at 16,000 pixels per second, sorry. And last but not least, this camera is optimized for low KV operations, and I'll show you an example of how this can be beneficial. Okay, so we use the Dexterous hybrid pixel technology in the Stela camera. What that means is, first of all, this is a counting detector. So you get minimized noise because of electron counting. And the second thing, as I said, you get the highest diffraction pattern, highest dynamic range, sorry, because they do on-the-fly digitization as they collect the diffraction images. And because of instant retriggering that is implemented in the detector, you can do diffraction imaging at higher dose rates than commonly used for electron counting detectors. So this is what makes this camera the best detector for diffraction imaging at low KVs. You have electron counting with minimal noise, you have high dynamic range, and you can go to higher dose rates, which are more common in diffraction applications, and collect your weak and high intensity diffraction spots with the highest possible signal to noise ratio. Here's an example showing you what I mean by two things: electron counting, which gives you minimal noise, and also high dynamic range. So this is a single, on the left-hand side, a single frame with 0.5 millisecond exposure time on a zinc oxide nanowire. And you can see here is the central beam, the 000 beam, and I have far out and high order reflection up here. And what I have done is I've drawn a profile line along this low intensity diffraction spot and also the central beam, and then grafted in log scale on the right-hand side here. The reflection up top here, you can see that here on the left-hand side, and you can see it has maybe 4 or 5 counts, but it has been detected. And on the other hand side, you see the central spot here, which has around 40,000 counts. So I have a spot with a few counts on the left-hand side, and I have 40,000 counts on the central spot here on the right-hand side. So I have been able to detect this very weak reflection, at the same time include my my high intensity 000 beam in my diffraction image. And this is a single frame with 0.5 millisecond exposure. If you do frame summation to collect a longer exposure time diffraction pattern, you actually multiplying your dynamic range as you're summing the frames. So this is just showing you a single frame example, and I can go and improve this in both the signal to noise ratio and the dynamic range as I sum frames. Let's say if I collect a 1 second image and I sum 1,000 frames like this. Here are a couple of other examples from diffraction images on different types of specimens. These are just parallel beam diffraction. A lot of times people ask us, are you not worried about damaging the sensor? Do you need a beam stop? So here are some examples of showing you that you do not need to use a beam stop with this camera. It's a very robust system. And you can see different examples I have here from gallium arsenide, tin oxide, and zinc oxide again. And the reason usually we invert the contrast in our diffraction images in some of our slides is to show you that it's a very clean, artifact-free diffraction pattern, as you can see here on the bottom. Now, as I said, the Stela camera, it has different operating modes, and you can run the camera up to 16,000 frames per second. And when you connect this to a STEMx, this means that the camera can drive the scanning probe at 16,000 frames, pixels per second, sorry. So you can collect 16,000 pixels per second, 40 STEM data with this camera. And that is done using a sub-area readout in the sample. And when you can collect 40 STEM data sets with a faster frame rate, what that means is you can cover a larger area of the specimen, as you see in this video that I just played, within a shorter amount of time. So you can have more statistics about your specimen as you're collecting your data. It also means that for beam-sensitive samples or specimens that drift a lot, you can minimize both the drift and damage on your specimen as you're scanning your sample. Here is an example of a 40 STEM data that is, again, everything is done in DM. You set up the experiment, you collect the data, and you process the data within the same software interface. These are some gold nanoparticles. So what you do is you specify your region of interest, you collect your 40 STEM data. You can see from this 40 STEM data, I have extracted virtual selected area electron diffractions using these selections, region 1 and region 2, and those are these two diffraction images on the right-hand side. And then this is the example where I told you we have used a Python code run in digital micrograph to generate this grain orientation map. And the way that the script works is it goes pixel by pixel. It looks for the highest intensity reflection in the diffraction pattern in that pixel, and then it classifies the pixels and color codes them based on the orientation of the highest intensity reflection. This is how you can get not a quantitative but a qualitative visualization of the orientation in your specimens in digital micrographs. And the other point that I made about the Stela camera is that this detector is optimized for imaging at lower KVs. So with our other detectors like the OneView, Rio, and the K3, the optimized operating ranges are usually above 80 kV. With OneView and K3, you can go up to 300 kV, and with the Rio camera up to 200 kV. But we normally do not recommend using these cameras at lower KVs, below 80 kV specifically. Stela, on the other hand side, is optimized for electron energies below 120 kV. So, and the sweet spot is within the lower kV ranges. So this is very complementary to the other detectors that we had before. And what this means is that you can actually use the Stela detector for material samples that are very, very beam sensitive. And I have some examples, so let's look at that. Molybdisulfide is a beam sensitive sample. And here is an example collecting a 4D STEM data and then using virtual imaging again, so to identify defects, interfaces, and rotations in a material like this. This data set was collected at Northwestern University. We have recently installed a Stela camera on their grand arm. And they are using the system, and they shared this data with me. So what you see here is virtual dark field images generated using the 4D STEM data here, and then the color map is generated basically using a combination of these virtual dark field images. The data was collected at 80 kV. To reduce the specimen damage at 2,000 frames per second. Each 4D STEM, the data cube contained 400 by 400 scan pixels and 256 by 256 diffraction images. So the second point I wanted to make is how easy it is to use the Stela camera and collect the data and process the data with the system because it is fully integrated in digital micrographs. So as you know, digital micrograph is an industry standard for different types of electron microscopy applications. You can do regular TEM imaging, in situ microscopy, tomography, and 4D STEM. And with the Stela camera, we have fully integrated 4D STEM and diffraction applications with a hybrid pixel electron detector, and that's the only hybrid pixel electron detector fully integrated in our software. What that means is instead of using a traditional workflow where you get on the microscope, you set up and optimize your experimental parameters, then you acquire your data, and you have to transfer your data and convert your data to a different format to be able to mine it and analyze it, and then maybe go back to the microscope to optimize your data acquisition parameters. And once you're done with data acquisition and optimization, you transfer and you manage your data. Everything is going to be done in one software interface. So with Stela integrated in digital micrographs, you can set up, optimize, acquire the data, mine the data right at the microscope, go back and maybe change your data acquisition parameters, acquire the data set, analyze your data, and manage your data in one software interface with one type of data, no data conversion needed. And in addition to existing data processing tools that you have in digital micrographs, as I showed you earlier, you can use DM scripting and Python scripting to do further analysis on your data sets. And all of this means that you have shorter time to results because everything is simply done in a single optimized software interface. Right, so this is an example where, for example, you do a DPC data acquisition, you process your data, and very quickly at the microscope, you decide if you want to go back and change acquisition parameters on the microscope, like I need longer pixel dwell time, or I want to scan a larger area, or I need a different camera length, things like that. All right, so this was a quick introduction to 4D STEM technique and also the Stela camera. I am now going to switch to my other computer, which is connected to the microscope. Okay, so you should be seeing my digital micrograph interface now. And we are going to, I'm going to give you a brief introduction to the software interface. We will collect some 4D STEM data, and then we will analyze some data in DM as well. All right, so GMS 3 is the new generation of digital micrograph from Gatan, which has been around now for several years. We made big changes and improvements compared to older versions of digital micrograph. On the left-hand side here, you can see the microscope system software interface. This is an interactive interface where I can control both Gatan hardware that are connected on the microscope to DM, and also the microscope itself. You can get information about the microscope mode that you're using, if you're changing the magnification, if you're in scanning mode or TEM mode. As I said, depending on what hardware you have installed, you will have different tools shown in the microscope display, and you can interact with them. On the right-hand side, we have the techniques page. This is, again, depending on the hardware that you have from Gatan and the software licenses that you have different techniques available to you. So we are going to do 4D STEM. So I have selected STEM-SI, STEM spectrum imaging technique. And when you go into a technique, when you make your selection, it gives you all the tools that you need to run an experiment for that technique. Let's say I want to do TEM imaging, or I want to do in situ microscopy. If I go to the in situ technique, it's going to give me all the tools that I need to collect an in situ data set. Now that I want to do a 4D STEM data acquisition, in the 4D STEM, in the STEM-SI technique, I have the DigiScan control, the SI acquisition window, and also the the selected camera interface where I can I can set up my camera parameters. So this is very workflow-based design. We have thought of running TEM experiments and STEM experiments simplified for the users. So if you start from the top of the window and you make your way down to the bottom. You start with setting up your parameters, data acquisition, and then data processing at the end. The other nice thing that I really like about the new GMS are these workspaces. And this is basically if you have, as you're collecting your data, if you have different types of specimens or different regions of the sample, you can put your data sets in these different workspaces. And then you can store these workspaces as DM workspace files. When you do that, with the workspace, the data within the workspace is going to be also saved. And when you open up your workspace later on, it will open up your data set the same way you had it organized last time you closed and saved your workspace. Yeah, and then on the left-hand side here, you have the regular display settings. Okay, so that's the introduction to GMS3. Let's start looking at how we use the Stela camera and how we collect the 4D STEM data set. This is the camera window to control the Stela system. As you can see, I have different modes: HDR, which is the high dynamic range mode, and high speed, or HS. And I also have options to select a readout area of the sensor: full readout, half ROI, and a quarter ROI. With a combination of this ROI and also the camera mode, I can change data acquisition parameters and frame rate of the camera. So with the HDR mode in full frame, I can run the camera more than 2,000 frames per second. And if I go to the high speed mode and I select a quarter ROI, this is how I can collect a data set with more than 16,000 frames per second. You can see I have the sensor size is 1K by 512. I have the option to crop this to a square if needed to, and I'll show you an example of this a little bit later. And this might be useful if you wanted to reduce the data size as you're collecting your 4D STEM data set. And so you have the live view option here, and also you can capture diffraction images here. The example that I showed you earlier in my presentation from a single frame diffraction pattern with the live profile going along the very weak reflection and the high intensity 000 beam. So I wanted to just zoom into here and see, here is the spot with just a few counts. And you can see if you look here at the bottom, as I scroll on my reflections, those are the number of counts in those pixels. And I can, as I told you, increase my dynamic range and improve the signal to noise ratio in my diffraction pattern if I do dose fractionation data collection. So here is my reflection again. What this image is a sum of a multiple of these single frames. As I sum them, I also increase my dynamic range and multiply my dynamic range by the number of frames that I'm summing. And that is done here using these options. So you put in the total exposure time that you want to use for your acquisition and the number of frames that you want to fractionate that total exposure time to. So if I say 1 second and 2,000 frames, each of those frames is going to be 1/2000 seconds, and then it's going to sum it and give me a summed image. I have also the option to save the stack, so have all the individual frames saved for me as well. Okay, so let me unblank my beam. I have I should have mentioned, I think Parker mentioned this earlier as well. We have a T20 here in our applications lab, and the Stela camera is mounted on the T20 with the lab 6 filament. And I am running the system in scanning mode, and I have a fib lamella sample inserted with a single tilt holder. So I was able to bring it close to zone, but it's not perfectly on the zone axis. So I apologize for that. And here is my STEM image. Let me start a camera view. So I'm looking in high dynamic range, full frame mode. This is my my park position for my scanning beam, and I am going to move my beam on the surface of the sample. So here you are looking at the silicon substrate, and I can move it and look at my diffraction image as I'm going through different regions of the sample. So to start, for a 4D STEM data acquisition, this is what you want to do. You start you put the microscope in STEM mode, you do all the regular alignments with the Ronchigram. You go to the region of the sample that you're interested in for data acquisition. Then you start a live view on your camera, and you make sure that your imaging parameters are as you want them. You can change the camera length, you can change the exposure time, the sub area readout, and all of those can be optimized for your data acquisition. Then we go to the SI acquisition window. I want to collect the 4D STEM data, so I select 2D array, and this green box represents the region of the sample that I want to collect the 4D STEM data from. So I can move this box, I can change the size. Here in SI acquisition window, I can put in how many pixels I want in this box and what is the pixel step size, so 5.4 nanometers in this case. And I can put in here the pixel dwell time. And this is equivalent to 2000 frames per second. So as I said, with HDR full frame, we can run the camera at this frame rate. So let's collect the data at that frame rate. There are a few other parameters you can change here. For example, if you wanted to do additional binning, so right now we have 1x hardware binning. So my images, my diffraction images are not going to be binned. But if I wanted to reduce my data size, as I am collecting it on the camera, I have the option to do additional software reduction. I will talk about drift in a little bit later, but you can also do drift correction as you're collecting your 4D STEM data set. This is normally not needed when you collect the data at high speed, but I'll show you how it works just in case you're interested in using that. And then there are some other additional tools here. For example, you can automatically blank the beam and close the column valve after data acquisition. You can have it automatically save the data for you once data acquisition is done. Okay, I think I'm all ready to collect the data. So we are going to scan a 128 by 120, let's put 128 area here, and it tells me that it's going to take 9 seconds, and the total data size is going to be 32 gigabytes. So I click on capture. You can see here it's scanning with the yellow line, the sample region that I was interested in. And as it's scanning, it's sending the data into digital micrograph. And here is my 4-dimensional data cube. Once it's done collecting the data, it automatically blanks the beam for me. So this is a 4D STEM data set. You can see here, I have 4 dimensions, or you can see up top here, I have 4 dimensions. So the first 2 dimensions are my diffraction image dimensions, and the second 2 are my real space 128 by 128 scan area. And if you use the picker tool by right-clicking on the image, I can use this as to look at the diffraction images in my data set as I'm moving over the sample. So here you can see some of the metals. All right, so this is collecting the data at 2000 frames per second, but I like to go even faster than this. So let me go back. What I want to do is collect a high-speed data set at a quarter ROI. And because my camera length is not that big, I can actually crop to square, so I don't need to collect the rectangular data set. This helps me reduce my data size. And with this selection for camera mode, I can run the camera much faster. Let's do that. So this is equivalent to more than 16,000 frames per second. And because I'm running this fast, let's improve my spatial resolution. So I'm gonna increase the number of pixels that I'm gonna use in this selected region. So my scanning step size is cut in half now because I'm scanning twice the number of pixels. And it's telling me that it's going to take 5 seconds to capture this image. So let's do that. Again, camera is going to be the timing master. It collects diffraction images at each pixel position. It is now done scanning the sample, and as it's scanning the sample, it sends over the data to digital micrograph. So this is my 4-dimensional data cube again, and I can use the picker tool to look at my my diffraction patterns. I could have centered the diffraction spot a little bit better. But yeah, here is our 4-dimensional data cube. Now, because I am scanning so fast, you can see a 256 by 256 scan took 5 seconds to capture, as you can see here. What you can do in some cases, if you do have drift or you have a beam sensitive sample, you can actually do a data acquisition with multiple passes. This is very common when you're doing spectrum imaging with EELs. You can do that, the same thing with 4D STEM data acquisition. So let's say I want to collect 3 passes. And what this means is it scans the surface of the sample at the maximum speed once, and then it goes and scans the second time and adds the diffraction images from the first round and the second round, and so puts the summed diffraction image in each pixel position. And it does it the third time. So the third time you will have per pixel a diffraction pattern that is a summation of 3 times acquisition. So this, instead of increasing my pixel dwell time by 3, helps with reducing damage on some types of beam sensitive samples. Now that we are doing 3 passes and it's still taking very short amount of time, let's also see how drift correction works. So I click on drift here, and I can select how often I want to do the correction. Since we're doing 3 frames, let's do it once a frame. So it captures 1 frame, and then it does a drift measurement, and then corrects it, and then captures another frame, and so on. And when I click on drift, it gave me this yellow box. This is the spatial drift box. So the way it works is you want to put the region on an area of the sample that you have enough contrast. You need to put in your STEM detector so that during data acquisition for each drift correction, it actually captures a STEM image as you're collecting the data, and it measures the drift and corrects for it. So let's start the acquisition. There is the reference STEM image. So it took 2 images and it cross-correlated them. Now it's collecting the first round of 4D STEM data. Then it's going to take a second set there. So this is the scan after the first scan and the cross-correlation result. And the second time scanning. And drift measurement and correcting for the drift, and the third time scanning the sample. All right, and done. So as I said, each pixel in this data set is now a sum of three diffraction images acquired on the surface of the sample. Okay, so this is how you collect the data. Let me close the call valve. As you can see, it's really, really easy. You put in the sample, you go to STEM mode, you make sure your STEM alignments are well, and you find a region of the sample that you're interested in, and then setting up and acquiring the data is very quick and easy. Now, let me show you how we process the data in DM. So the first example, I want to talk about virtual imaging. As you saw, I can use a picker tool, and this picker tool would be a virtual selected area electron diffraction aperture. And as I move my picker tool on different regions of my sample or different areas in my data set, you can see I am getting the virtual selected area electron diffractions on the right-hand side. So this is having a virtual selected area aperture. If I go on the diffraction domain, I click on range, show range, and you can see this green slice box. And what this is a virtual objective aperture. So by moving my objective aperture around, either on the 000 beam, I can get a virtual bright field image displayed here, or if I move it to one of the reflections, I get a virtual dark field image. And you can see as I move it around, different regions of the sample, depending on their crystalline orientation, they light up. The shape and size of these objective aperture or selected area aperture could be any shape and size. You can use different ROI tools to draw and design your apertures, but you can also use one of the masks here. For example, this bandpass mask, I can put it on and generate a virtual dark field detector, and then calculate the signal using this virtual detector. There, that's my dark field image from my virtual dark field annular detector. Okay, we are almost running out of time, so I'm going to quickly also show you how we do strain mapping. In order to do that, I'm going back to the technique page, and in data analysis, I'm going to select 4D STEM, and here is the strain mapping window. As I said, the way strain mapping works with nanobeam electron diffraction data sets is we compare the diffraction patterns in the data set with a reference diffraction pattern. This is a silicon germanium multi-layer specimen. So on the top, I have silicon germanium, silicon germanium. As you go down, the content of germanium increases in these layers. So you would expect higher strain as you increase the content of germanium. The first thing you want to do is to specify in the software an unstrained region. So that is using this tool, and as I'm showing here with the unstrained box. Once you have that specified, you basically follow from top to the bottom of the window. We first want to calculate the reference diffraction pattern from that unstrained area, and you saw in the presentation, using this reference diffraction pattern, you specify the directions for which you want to calculate the strain, so the growth direction and perpendicular to the growth direction. This green box allows you to select how many orders of the reflections, if you want to include higher orders in your calculations or not. Of course, if you do have enough signal to noise ratio in your higher order reflections, it's better to include them in your calculations. Once you have specified your directions and you made your selections, you click here on disk, and it is going to calculate a template this template to use to find the position of the rest of the reflections in your diffraction data. And then you're all set and ready to calculate your strain map. And here you have an option to select a sampling interval. And this would be useful, let's say, if you have collected a 512 by 512 scan area and you're at the microscope and you want to quickly calculate the strain map to make sure you have the quality of the data that you needed, or you need to adjust the parameters. You can calculate with a subsampling, let's say every 5th pixel or every 2nd pixel. This reduces the calculation time and gives you a quick map in order to quickly assess the data quality in your results and go and modify it on the microscope if needed. I'm just going to keep it to 1 for now. And when I click on calculate, it is now calculating a strain in the directions that I have selected. The 3rd window is shear, and the last window is the rotation. So let me quickly set this up into a color and set the contrast limit. And you can see here, so I'm setting up the same limit for both X and Y strains. And you can see I'm calculating, or I'm detecting strain in the silicon germanium layer, and as it goes down, the strain is increasing. As I said, there is more germanium as I go down in the layers in this specific sample. And we didn't find any strain as expected in plane direction. Okay, in the interest of time, I am going to stop this one. And my last example to go over is how we do differential phase contrast imaging in digital micrographs. Here is an atomic resolution 4D STEM data with a large convergence angle. You can see as opposed to the data set that we used for strain mapping, which was using a small convergence angle close to in nanobeam diffraction, this one is collecting only the 000 beam using a large convergence angle. This was done with a K3IS camera at the end of a GIF, and the data was shared with me from NSEM at Berkeley. And so what we want to do for DPC analysis is measuring how much the center of mass is moving in our electron beam diffraction and correlating that to the electric field in the specimen. There are different ways to do that. Conventionally, people used to have physical segmented detectors on their microscope, and by measuring the signal on different segments on their detectors, they would measure how much the beam is getting deflected, and then from that calculate the electric field. Because we are collecting the full diffraction pattern at each pixel position using 4D STEM, we can do that virtually. So here I have created a mask with 4 annuli. This one that I have created before has 4 segments and 1 annulus, but you can have different selections and create your own mask. I will show that mask. I will show that mask here on my diffraction pattern. I can edit this mask. I can change the diameter, the inner and outer the diameter of the mask. I can move the segments around and change their size, depending on how you want to do the calculation. And then once you are done editing your mask, you can go in and put in different formula for signals that you want to calculate using this virtual segmented detector. And once you have designed your also signals, you can go ahead and have the software calculate these signals for you. So there we go. I had specified A minus C and D minus B before. So the software is now calculating these two signals based on this virtual segmented detector that I designed. Now that I have these two signals, I can combine them and calculate my beam displacement vector map, and from that, the electric field in the sample. Another way to do this is instead of just using segmented detectors, I can actually look at the full diffraction data and calculate the center of mass shift. So let's do that as well. I don't need to do anything. I just select center of mass and use full area, and I click on measure. Oh, I need to have this guy in the front. And the software is going pixel by pixel, calculating the center of mass in that diffraction image, and giving me the shift in the center of mass position with respect to a reference center of mass in X and Y directions. Now that I have these two components, I can use these two to generate my beam displacement vector map. And I do that, as I said, you just go from top to the bottom of the window. So here is the vector displacement. I click on calculate. It asks me to select my X and Y components, so dx and dy for X displacement and Y displacement. And this is my beam displacement vector map. So the color here shows the direction of the vector, and you can see this wheel that tells you which direction that corresponds to. And then the intensity at each pixel is the magnitude of the vector at that pixel position. So this is a couple of different ways that you can do DPC also in digital micrographs. This is all I wanted to show you for today. I went a few minutes over. I don't know if there are any questions, Parker.
We have a lot of questions, but
okay.
Again, we are out of time.
Yes.
Are you willing to answer one?
Sure.
Okay. This is I'm starting from the beginning. So, is it possible to implement Stela camera and STEMx into a GIFT quantum system and into a TEM system operated by Windows 7?
That's a very specific question. Let me get back to you. But Stela is not compatible with the quantum. It is compatible with our newer generation of GIFT Continuum. And we just announced that this week or last week at the PICO conference. So Stela can be mounted at the end of a GIFT Continuum, and you can do filtered 4D STEM diffraction data acquisition with a hyperpixel detector at the end of that system. Yes.
I thought that was an appropriate question for today. So
yeah.
With that, I want to thank you very much, Anna. It was a great workshop. If anybody has any more questions, please put them in right now and we'll get to them in the next week. And thank you, everybody, for attending.
Thanks, everyone, for your attending.